Thanks, everyone. When I started putting this together, realizing that I only had 10 minutes to cover the biology of an entire family, I tried to basically condense what was known for sawfish in the relevant aspects of what the focus of the workshop is, as we talked about earlier today. So what I decided, and actually backing up for a second, I just want to acknowledge the person who works in my lab, Kelsey Smith. This summary that I'm going to provide to you is part of this database that Lucy had mentioned early on that we've been compiling over the last year to try to have basically at our fingertips, information about the biology of the family of the sawfishes. So I just want to acknowledge all of her hard work for doing a lot of the data mining and the digging. So realizing that Vicente was going to cover genetics, I could basically put that one aside. But then biology, I kind of took some major groupings, and some of you may agree or disagree with this list, but there are obviously other aspects of people's talks that might fill in some of these gaps of the biology of sawfish that we can discuss as we move forward to this workshop. But for the purpose of this talk, I just want to cover some of the major areas of interest which we would consider biology of the animals. And first, the habitat use and migration, which will obviously have some consideration as we move forward, looking at threats and constraints of the animals. Of course, some of the general life history information, such as age and growth and reproduction, which ultimately use in population models to predict the vulnerability of their to extinction and the ability of them to recover. Dieting, feeding, and predation are they are related to their life history and their biology, and it would just be interesting to note that. And then lastly, as I said, the demography, which relates back to the age and growth of the animal and the reproduction. So my apologies to anyone that I may have missed as far as documenting their work or their paper, but as I said, in 10 minutes I've tried to put as much information as I could to try to give a general overview of everything that's going on that our current knowledge for this family is. So obviously there are going to be exceptions, because a sawfish in Australia may do something very differently than sawfishes in the United States or in Africa. But for this talk, I thought I'd kind of find some generalizations that we could use that would help us in our goal as we move forward. The first, and obviously falls out for many of you in the room, is most of the species, the data that we have collected indicates that they're largely found in euryhaline environments, but they do move between estuarine environments and marine environments. There does appear to be, from telemetry data and from encounter data, areas where the animal was reported, positive associations with mangroves and seagrass habitats and with muddy bottoms. And for anybody who's ever been out trying to track or find sawfish, the mud is not just mud, it's mud. Meaning you step into it and you look to your waist. But obviously, as I said, there are going to be some interesting exceptions. And one of these we started looking at is freshwater encounters. I mean, these would be animals strictly found in a freshwater environment. One would be Pristis pristis, the sawfish formerly known as periateti, that was documented in Lake Nicaragua and some of the freshwater areas in Central America. There are also reports of clavata in rivers, moving far up rivers in Australia, and also of microdon found in freshwater billabongs in Australia. So in general, the species does have a very high tolerance for salinity, found in 0 parts per thousand and then all the way up into marine environments.
What's billabong?
A freshwater pond, I guess, be the best way to describe it, Colin?
Billabong is usually a bend in a river that during the dry season will retain water when the rest of the river is dried out.
So, thank you. Along the lines of habitat use and migration, there also appears to be a shift in their movement patterns. It shouldn't be too surprising for some of us that work on animals, is that generally the smaller animals are limited in their movements just simply because of their size. Larger animals are generally found in deeper water. Larger juveniles generally have larger home ranges, so they occupy more activity space in a given period of time, usually daily. Some acoustic tracking work that was done by Colin and some others found that individuals generally will move along the shoreline, the mangrove shoreline. And they tend to move with the tide, moving out with an outgoing tide and moving back in an incoming tide. There also seems to be some affinities, although there can be some exceptions to the rule depending on which species we're looking at, with the abundance of animals. They tend to have an affinity for very shallow water, which would explain, obviously being found around shorelines. Generally more warmer waters, over 30 degrees centigrade. The dissolved oxygen is not too surprising. generally finding areas that are not anoxic is a good thing, or hypoxic, but generally above 6 milligrams per liter. And then a salinity, which you can see here is very broad, 18 to 30 parts per thousand. And I have a feeling if some of these tracking studies were done in more euryhaline environments, we may see salinities even further lower than that. Now, as I said, most of the data that we have is strictly because these animals are more available to us. We've been able to capture them. Most of this habitat data that we've collected is on juveniles. We still have a long way to go of what's going on with the adults, especially on their long-term movement patterns and habitat use. Is that a fire alarm? What was that?
I think it's just a test.
Ah, great. Right in the middle of a good old fire alarm. Awesome. They got a good 10 seconds, yeah. So as far as the adults go, this is a big gap that we have. There's still a lot of information we need to collect on the adults. I mean, when we go back and look at the historic records, we do see, animals that seasonally may have made large distances in movements. However, we have some of myself and some of my colleagues here in the room have been putting satellite tags on some larger individuals in the United States, and we're finding that they're really not moving that far. What that means in the big picture, we don't know, but it's just something that's interesting is that historical records indicate big movements, but we haven't seen it so far with the satellite tracked animals that we have data on. Now, taking another step, and just for the good of the order, in terms of age and growth, for those that are not aware, we can determine growth of an animal by several different ways. One of them is by simply tagging the animal, tagging it, and then recapturing it, and then just measuring the growth from when it was initially tagged to when it was recaptured. All right. We can also fit growth models to the data. We can fit growth models to our tag recapture data. We can fit growth models to our length frequency information. And a very common way to do it is to fit growth models to growth bands. Again, just for those that are not aware, what we do is, this is a section of a vertebrae from a sawfish. And very similar to rings on a tree, we can count the bands. And we take the number of those bands and we assume that the number of bands formed is related to their age. For example, a sawfish having 3 bands potentially could be 3 years old. You can spend a whole talk about this, but just for the good of it, we generally just count bands on vertebrae and use those to determine the age of the animal and fit that to a growth model. So we've had some success over the last few years looking at age and growth of these animals. But just to kind of give you an idea, is the early estimates that were done for these animals tend to indicate very, very slow growth. This K is the growth coefficient, generally a higher number indicates the animal grows faster. So you can see this value is very low, 0.06, which really indicates the animal grew slowly. This was a study that was done by Tanaka on Priscus microdon. The growth equation also gives us a theoretical maximum size, how big the animal should be getting. And then also a maximum age that was determined to be about 40 years. So very similar to a lot of the elasmobranchs, we assume that they have very slow growth and long life. Also, Colin did some work on some data that was collected by Thorson and also came up with some similar, very slow, long-lived animals for Periacaete, or now, Priscus priscus. However, over the last couple years, we've seen some interesting shift in that we don't think they grow as slow as we initially thought. There's been several growth studies that have been done during a bunch of different ways on several different species. Note here, this is a study that was done by Peverell. There was some growth for microdon that was still considered slow, but if you look at cuspidata, quite a bit higher. Clavata, slow. And Zistron, quite a bit higher. In the United States, 2? I thought I got a little bit longer because of the fire alarm. Okay. In the United States, we're also starting to see some work with using tag recapture information and from fitting size at age data, and we noticed that the growth coefficients are much faster than we originally thought. And if you look at this growth model fit over the data, this is from one that was fit to size at age data and one that was fit to tag recapture. They look very similar through the early ages, and the only real difference is that this had larger animals, which would cause the curve to fall down, and this had, you didn't have any larger ages, so there's a bit of a disparity there. But the important thing is that 2 separate methods showed very similar fast growth during the early ages. Now for reproduction, again, most of the data that we have is anecdotal. back towards the slow growth, long longevity, with females maturing at very late ages, up to 25 years, although we think it's an overestimate. But in some cases, now it looks like it could be around 8 to 10 years. Fecundity, how many pups they have, it's all over the place. For those of you that have never really seen the inside of a sawfish before, we were lucky enough last year to have an animal that washed up due to a cold striking event. And this is a dissection from a large adult female. And these are all the oviducts, the eggs. And when you cut it open, I mean, these were the size of cantaloupes. They were very, very large. It was sad to see a large animal like that die from cold stunning, but it provided a lot of information for us. In fact, this was the first adult female that we've looked at in the United States in about 15 years. Diet and predation, again, a lot of anecdotal data. Small fishes, the predators, crocodiles, sharks. Now, lastly, I just want to cover one final thing is the demography. Again, some early stuff that was done on the demography and how fast the population grows was using those old growth estimates that I was talking about. And what that indicated is the population was only growing at about 8 to 10% per year, which was pretty slow. And even slower for Pristis pristis, about 5 to 7%. But using some of the new values that we have, and especially that growth coefficient K, which is also used in estimates of natural mortality, if we take that and kind of do a back-of-the-envelope little calculation, or more than a back-of-the-envelope, I've been working on this for the past couple months. If we look at using an age of maturity of about 11, a maximum age of 30 years, and a much faster growth, it results in a much faster growth of the population than what we originally thought, which is good news. And what that ultimately means is that this will help us when we go back and revisit some of those early population viability estimates and the risk of extinction, because the populations we now know are growing a lot faster than we originally did. But as you can see from the brief talk that I've given here, we still have a lot of data to collect. And another big thing we always get asked as scientists is how many sawfish are out there? And I don't think we really have a good idea of that. I've heard estimates anywhere from 2,000 animals to 15,000 animals. It seems like it's all over the place. So I will end there.